All right, so this video is about the light-dependent reactions of photosynthesis, and this is kind of the start of how plant cells and chloroplasts, or any eukaryotic cell with chloroplasts, makes its energy. So photosynthesis is divided into two steps, and so we'll break this video into two parts. The light-dependent reactions and the Calvin cycle. So in the light-dependent reactions, that's where we find something called the electron transport chain, and we'll describe that a little bit further in this video. And then in the Calvin cycle, that's where a process called carbon fixation occurs, and we'll discuss that in the next video. So there are three main goals of photosynthesis, and in the light-dependent reaction, the first two goals are to capture light energy and to make chemical energy. And that chemical energy is in the form of ATP, which is cellular energy. That's what all cells use as their cellular energy. And then NADPH, which is something called an electron carrier. And animal cells have electron carriers as well, but in the chloroplast, NADPH is the electron carrier. Now the Calvin cycle, its job is to make glucose, and glucose is the food for the plant. So when the light disappears and they can't make ATP energy with photosynthesis, with the light-dependent reactions, then they've produced all this glucose in the Calvin cycle, which they can then use in their mitochondria later on. And it also helps to make other organic compounds. The Calvin cycle actually makes a different molecule called G3P, which we'll talk more about in the next video. But it can use G3P to make glucose, but it can also use it to make other organic compounds in the cell. So just a reminder that there are these light-capturing pigments in chloroplasts. And we know them as chlorophyll. Chlorophyll is, here's the chlorophyll molecule here. And remember, it's got this magnesium ion at the center of this carbon and nitrogen ring. And then it's got this lipid tail that sticks into the thylakoid membrane, the phospholipid bilayer of the thylakoid membrane. And this magnesium ion is responsible for absorbing photons of light, little packages of light. At certain wavelengths. And remember that it prefers to absorb the blue-violet light and the orange-red light of the visible spectrum. And so what we need to know is that, again, that lipid membrane is stuck into the thylakoid membrane, into this hydrophobic interior, because lipids like to hang out with other lipids, or nonpolar things like to hang out with other nonpolar things. So it sticks into the hydrophobic interior of the phospholipid bilayer of the thylakoid membrane. And then the magnesium ring is facing the stroma. Remember, the stroma is the space that is inside the inner membrane of the chloroplast, but outside the thylakoid membrane of the granum, of the grana. And those electrons, the electrons of the magnesium that belong to the magnesium atom, they absorb photons, and they actually jump off of that magnesium and down something called the electron transport chain. So this is really responsible for the flow of electrons. There's a lot happening here in this magnesium that that are really important for the process of the light-dependent reactions, especially the electron transport chain. So here it is. Here are the light-dependent reactions. And the light-dependent reactions are kind of broken into a few parts here. So we've got photosystem 2, we've got photosystem 1. Here's the electron transport chain that's between the two photosystems. And then at the very end here, we've got an enzyme called NADP plus reductase. And so you can see here in the photosystems contain the chlorophyll pigments. And so here's the chlorophyll pigments, and they're on the stroma side of the thylakoid membrane. As a photon hits these pigments, the yellow arrows are showing electrons bouncing from one to the next to the next to the next. So let's talk a little bit about what happens at photosystem 2. So let's talk about these things individually. So at photosystem 2, what's happening is that chlorophyll will absorb a photon of light. And what that does is it causes the electron of that chlorophyll pigment to jump off. The electron gets so much energy that it jumps off of the chlorophyll molecule, and it bounces down into photosystem 2. And photosystem 2 gives that electron pair to the electron transport chain. Now, the chlorophyll molecule that had the electrons knocked off of it needs to replace those electrons. And so those electrons are replaced by water. So remember that one of the requirements for photosynthesis is water. One of the reactants is water. And so this is where water is used. It's used at photosystem 2. Water is split into its components. And so you can see the formula up here. Water is split into 2 protons, 2 electrons, and an oxygen atom. And the electrons from the water molecule, they don't just float around in the thylakoid space. They actually are the ones that replace the chlorophyll, the electrons on chlorophyll. And then light will hit that chlorophyll again and knock those electrons off, and photosystem 2 will split another water to get new electrons. So water is kind of like the electron input to photosystem 2 and the electron transport chain. So all of the electrons that are moving through the electron transport chain came from a water molecule. Often you'll see the equation written as water is split into 2 electrons and half of an O2. In other words, that's just one oxygen atom, half of an O2. But that oxygen will not last, that oxygen atom, that single oxygen atom will not last very long in the thylakoid space because another water will get split almost immediately. And so usually what happens is that 2 water molecules, we can represent that equation as 2 water molecules are split into 4 protons, 4 electrons, and 1 oxygen molecule. So it takes 2 water molecules to create 1 oxygen molecule that we end up breathing in from the atmosphere. So remember that the oxygen atom the oxygen molecule, O2, is able to just diffuse directly across the membranes, right? Across a phospholipid membrane. And so that's just going to move from high concentration to low concentration. So that's just going to move from the thylakoid space, through the thylakoid membrane, out into the stroma, through the inner membrane, through the outer membrane of the chloroplast, and then through the cell membrane of the plant cell and out into the atmosphere. Out through the stroma of the leaf and then into the atmosphere. So that could be a really good longer answer question: is track the path of an oxygen atom from the thylakoid space to the atmosphere. Oh, I should also mention that these protons here, the hydrogen ions that are split off of that water are also really important. They actually start to build up in this thylakoid space and create a high concentration of protons inside of the thylakoid space. So the high concentration of hydrogen ions is coming from the 2 hydrogen atoms that get split off of the water molecule at photosystem 2. So photosystem 2 is responsible for producing the oxygen that we breathe, and it's responsible for creating this high proton concentration inside of the thylakoid space, and it's responsible for donating electrons, or I guess absorbing light, which gives the electrons in photosystem 2 energy, which can then be passed down the electron transport chain. Okay, so let's talk a little bit about the electron transport chain. The electron transport chain is essentially a form of energy, or I guess it's a way of creating energy. So if you think about like the way that electricity is transported through power lines, it's kind of the same idea. And so electrons move from photosystem 2 to this electron carrier here. It's called PQ. You don't need to know what the name of it is. It goes through this, this protein here, this integral protein. It's called cytochrome complex, but what you need to know is that it's essentially a proton pump. And then the electrons get passed on to PC, which is just another electron carrier, and then they get given to photosystem 1. So electrons go from photosystem 2 to an electron carrier to the proton pump to another electron carrier to photosystem 1. That's all you need to know. You don't need to know PQ, PC, cytochrome complex. You don't need to know that. But what these do is that as they move from one electron carrier to another to photosystem 1, what they do is they lose their energy, but that energy is used to bring protons from the stroma to the thylakoid space. Okay, so that the energy from the electrons, the energy from the electrons is used to bring protons, hydrogen ions, from the stroma, where there's a low concentration of hydrogen ions, to the thylakoid space, where there's a high concentration of protons. Okay, so this further increases the amount of hydrogen ions that are inside the thylakoid space. The job of the thylakoid is to contain or create this environment that has a really high hydrogen ion concentration, because that's going to be important later. Okay, now let's talk about photosystem 1. So the electrons, let's just recap a little bit here. The electrons have come from water. I guess electrons have jumped. Photosystem 2 has absorbed light, which has given these electrons energy, which has now been passed down the electron transport chain. Those electrons have lost all of their energy, and now they need to get re-energized. They need to get recharged, essentially. And so photosystem 1's job is to take these electrons that have low energy, absorb another photon of light, and re-energize or recharge these electrons. And those electrons, they get passed on to an enzyme here called NADP plus reductase. And its job is kind of in the title here. The job of NADP plus reductase is to reduce NADP plus to NADPH. So what's going to happen is NADP plus, which is oxidized, is going to get the 2 electrons that come from photosystem 1, and it's going to collect 2 protons from the stroma, and it's going to help put those all together in a synthesis reaction and produce NADPH. So it's also using, so look at this, it's using protons from the stroma, which is decreasing the concentration of protons in the stroma. So it's further decreasing the hydrogen ion concentration in the stroma, making an even lower concentration of protons in the stroma, so that the concentration gradient between the thylakoid space and the stroma is even greater. This is awesome. This is all working out. Now this NADPH gets to go to the Calvin cycle, which we explore later in the next video. So this is the slide about photosystem 1. So again, those electrons that have low energy, they need to get recharged. So photosystem 1 absorbs a photon of light. That energy is used to recharge the electrons. Those electrons get donated to NADP plus reductase. NADP plus reductase takes NADP plus, which is oxidized, and 2 protons and the 2 electrons from the from photosystem 1, and it squishes those all together in a reaction called a synthesis reaction to produce NADPH, and that NADPH is used to power the Calvin cycle. Now, I've been talking about this concentration gradient between the thylakoid space and the stroma. And it's really important to understand that, it's really important to understand this. The thylakoid uses this high concentration of protons inside of the thylakoid space to power an enzyme called ATP synthase. So this is ATP synthase down here. It's kind of all alone. And so the protons are following the dotted red arrows, and they are flowing from high concentration to low concentration. So they're flowing along their concentration gradient. And the movement of these charged ions through this channel of ATP synthase powers the resynthesis of ATP, or the regeneration of ATP. So ADP and inorganic phosphate, so ADP, adenosine diphosphate, and just a free-floating phosphate molecule, there's plenty of these. These are in abundance in the stroma. These are in the plant cell. All cells are using ATP all the time to break, and they break it down to ADP and phosphate. They're doing this all the time. And so there's plenty of ADP in the cell, or in the stroma in this case, and there's plenty of inorganic phosphate. Remember, phosphate is PO4-3-. It's a molecule. It's a polyatomic ion. And ATP synthase job is to put those things back together. ADP and inorganic phosphate get put back together in a synthesis reaction to make ATP, adenosine triphosphate. Now it requires 4 protons going through ATP synthase to produce 1 ATP. So we do need to have a really good concentration gradient here, right? So the more protons that we can add to the thylakoid space and the more protons that we can remove from the stroma, the better. That makes a larger concentration gradient. Without this concentration gradient, then ATP synthase would not be able to produce ATP. The cell would have no energy, and so the cell would cease to function properly, would stop functioning properly. This is just kind of a simplified diagram of the light-dependent reactions. So here is the photosystem 2, and it's got all these chlorophyll pigments. Light is absorbed by chlorophyll, and what that does is it knocks the electrons off of chlorophyll and donates them into the electron transport chain. The electron transport chain uses the energy from those electrons moving from photosystem 2 to electron carrier to the proton pump to another electron carrier down to photosystem 1. So as the electrons move from one thing to the next in the electron transport chain, they lose their energy, but that energy is captured and used to power protons into the thylakoid space. Then those low energy electrons need to get recharged, so photosystem 1 will absorb another photon of light. Electrons get knocked off of photosystem 1 and sent to NADP plus reductase. This is another little electron transport chain, but they get sent on to NADP plus reductase. NADP plus reductase changes, or I guess regenerates NADPH. So it reduces NADP plus to NADPH, right? NADP plus is an oxidized molecule, and it needs to get reduced to NADPH. And so those electrons from photosystem 1 get used by NADP plus reductase. So 2 electrons plus 2 protons plus NADP plus gives us NADPH plus a proton. Okay, this is just an example of the cycling of a simplified version of the cycling of NADPH. Okay, remember that the electrons that NADP plus is going to get are taken from water lost at photosystem 2. They've come from the electron transport chain, right? And so the electron transport chain passes those down to photosystem 1. Photosystem 1 recharges them and puts them, or gives them to NADP plus reductase. And NADP plus reductase, sticks all of these things together. A proton, 2 electrons, gives us an NADP plus, gives us NADPH. This is the reduced form, and it's fully charged. It's ready to go and give those high energy electrons to the Calvin cycle. So now going the other way, NADPH can be oxidized to NADP+, which is the oxidized form. This is also the low energy form. Those electrons are then donated to the Calvin cycle, and that drives the synthesis of G3P, which we'll talk about later, and ultimately the formation of glucose. This hydrogen ion contributes to the, I guess can be, yeah, can go back to the the thylakoid space, and it would do that by going through the proton pump. Because remember, this proton here has been lost outside of the thylakoid space. So this proton is probably in the stroma, and so the stroma is going to need to put that back, or this proton needs to get back to the thylakoid space, and it'll do that through the proton pump of the electron transport chain between photosystem 2 and photosystem 1. Remember that these protons, these hydrogen ions, are abundant in the chloroplast. So these are just floating around, and they're easy to stick back together. All right, that's it, you guys. I know that you guys are probably going to have questions. I'm happy to answer those for you guys. Some things that you can do are just make a simplified diagram of the light-dependent reactions and just draw this over and over. Make sure that you've got all these important parts here. Make sure that you're describing these important steps. You should also be able to label the parts of the chloroplast, and you should also be able to label the parts of the leaf. Okay, now be careful here. There's a sneaky little trick question in this one here. You have to really pay attention. Pay attention to this diagram. Be very careful of where the structures are on this diagram. The spongy, we talked about mesophyll cells, but we didn't talk about the different kinds. The spongy mesophyll cells are the ones with lots of space. They look like a sponge, and that's to let the oxygen and the nutrients through and the water through. The palisade mesophyll cells are the ones that are near the top side of the leaf, right? So definitely make sure that you notice that. Okay, and that's all for you guys. If you have any questions, please let me know. I'd be happy to answer them, and I hope that that lecture was helpful.